First, we'll start by overviewing glycolysis and gluconeogenesis. And we're going to look at how aldolase is involved in both of those reactions and the different forms, isoforms of aldolase. And then examine how aldolase B expression is regulated by transcription factors. Let's review cellular respiration. The first step is glycolysis, then pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. In glycolysis, you have to spend energy to obtain energy. It's like telling someone who owns a business, you need to spend money to make money. First of all, glycolysis literally means the splitting of sugars. So we're going to take a 6-carbon sugar and we're going to split that into 2 molecules of 3-carbon sugars called pyruvate. This can occur with or without oxygen, and at the end of the reaction, we're going to get 2 pyruvate, 2 ATP, 2 NADH, and 2 molecules of water. And over here, you're going to see each one of those steps. And in each one of those steps, you're going to see a enzyme that's going to help go to the next reaction. And that's how we're going to learn glycolysis. It's by looking at the enzymes and its function. Okay, step 1 of glycolysis uses the hexokinase to phosphorylate or add a phosphate group to the glucose. The phosphate group is going to come from ATP that was added to start the process. Step 2 is going to isomerize the glucose 6-phosphate into a fructose 6-phosphate. And remember, it has the same molecular formula, but it has a different shape. Moving on to step 3. So at this step, there's another ATP molecule to transfer the phosphate group to form the 1,6-biphosphate from fructose 6-phosphate. Now at this point, 2 ATPs have been used. Look at step 4, which is the aldolase. It's going to then split the 1,6-biphosphate into a ketone and an aldehyde molecule. And you'll see them down here, the ketone and the aldehyde. And these sugars are going to be DHAP and GAP. Okay. Going to step 5. Step 5 is going to convert DHAP into GAP. And these isomers can go back and forth and interconvert. And then GAP, it's going to be used for the next step of glycolysis. So in step 6, we have the GAP being dehydrogenates. First, there's going to be the dehydrogenating GAP by transferring one of its hydrogen molecules to the oxidizing agent NAD to form NADH plus H plus the hydrogen. So next it's going to be GAPD. It's going to add a phosphate from the cytosol, and so then we end up getting there 1,3-biphosphoglycerate. So step 7, this next enzyme is going to transfer a phosphate from BPG to a molecule of ADP, which is going to give us ATP. This it's going to happen to each molecule of that, and that reaction is going to yield 3 of the 3-phosphate glycerates molecules and 2 ATP molecules. In step 5, it's going to relocate the phosphorus to 3 PAG molecules from the 3rd to the 2nd carbon to give us 2 phosphoglycerates. Now in step 9, the aldolase is going to remove the molecule of water from the 2-PGA to form PEP. And that's going to happen for each one of those that came from step 8. And then step 10, it's going to move the phosphate, the pyruvate kinase is now going to move that phosphate from PEP to ADP to give us the ATP. And that happens for each molecule of PEP. So at the end of it, this reaction is going to give us 2 molecules of pyruvate and 2 ATP molecules. Let's review one last time the parts of glycolysis. And the first step is the conversion of the glucose, D-glucose, into glucose 6-phosphate. So we know that this ring is phosphorylated and we have ATP. And we're losing that phosphorus, so now we get ADP. So that second step is where we have our next enzyme. And there's a rearrangement of the glucose 6-phosphate to the fructose 6-phosphate through the enzyme phosphoglucose isomerase. Then next to our next step to get to our fructose 1,6 biphosphate, we are going to use magnesium as a cofactor. And at the end, we have an ATP. And so then we're going to get fructose 1,6 biphosphate. And that next step, that's step 4, we have our aldolase. And it's going to split that fructose 1,6 biphosphate into 2 sugars. Now these sugars are isomers of each other. Remember, an isomer has the same molecular formula, but it has a different shape. And those 2 sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which is DHAP, and GAP. So DHAP and GAP. So what aldolase does there is catalyze the cleavage, and it yields 2 of those 3 common 2 molecules. Now we're going to that next step, step 5. In step 5, we're just going to use that enzyme, and it's going to convert the molecules, and the glyceraldehyde phosphate is removed so that it can go to the next step. When it goes to the next step, it's going to use the dehydrogenase, and it's going to add inorganic phosphate to the glyceraldehyde 3-phosphate, and it's going to produce 1,3-biphosphoglycerate. Now in that step, 2 things are going to happen. So the GAP is going to be oxidized by a coenzyme, and that coenzyme is NAD. And at the second part of that, it's going to be phosphorylated by a free phosphate group. And now we're going to go to the next step, which is step 7. And we see we have those 3 carbon sugars there. And at that point, it's going to form 1,3-biphosphoglycerate. Into step 7, we're going to have the enzyme that's going to transfer that phosphate from BPG of ADP to form ATP. Okay, remember it's going to form ATP. Now we're getting to the end of it. We're coming to the end, so we now have step 8, and that enzyme is going to relocate that phosphorus to 2,3-PGA molecules from the 3rd to the second carbon, and then enolase is going to come there, and it's going to remove a water. So when it removes that water, it's going to form PEP. And then that last step is going to transfer the phosphorus from PEP to ADP, and then it's going to form an ATP. And remember, we're going to have a net gain here. So I hope you understood all that word salad. Of course, there are notes that are left. There are notes that you can look forward to seeing as we go on. So you have two representations of the free energy changes and glycolytic reactions in your erythrocytes. So first you have the table that explains all of the free energy in each reaction, and then there's a graphical representation on the right side that explains how much energy is in and how much energy is coming out. So you want to lose weight. So how are you going to do that? You're going to do that by hopefully making your body use gluconeogenesis. So if we want to lose weight and we get on that keto diet, we're trying to force our bodies to burn the fat that we have stored. And that fat is going to have to go through gluconeogenesis instead of using carbohydrates that we were taking in, because now we're not using those carbohydrates anymore. So we're going to have to use that fat to burn for energy. Okay, gluconeogenesis is not a reversal of glycolysis. Now, I found a lot of references or a lot of information that says it's a reversal of it is a reversal. But the recent literature states very significantly that it is not a reversal. The reaction is to turn pyruvate into glucose. And gluconeogenesis occurs mostly in the liver. And we need this because at no time do we need to be without glucose. Of course, we know that our muscles need glucose, but what about our brain? Our brain needs very large amounts of glucose. So the steps of gluconeogenesis is on the side, and I found this mnemonic that is really silly, but it will help you remember all of the parts of gluconeogenesis. And so I want to go into each one of these in detail and talk about it a little. Gluconeogenesis can be explained in three steps. So the first step is the conversion of the pyruvate to phosphoenolpyruvate. And the next step is the conversion of fructose 1,6-biphosphate to fructose 6-biphosphate. And the third step is the conversion of glucose 6-biphosphate to glucose. So let's go back to that first step. So in that first step, remember, we're going to be starting with the pyruvate. So the pyruvate has to first be converted to oxaloacetate. And this is going to be done by pyruvate carboxylase. The oxaloacetate then has to be decarboxylated by phosphoenolpyruvate carboxykinase to form the phosphoenolpyruvate. Now, this reaction is going to require GTP. The phosphoenolpyruvate is then converted to fructose 1,6-biphosphate, and this looks like the reversal of glycolysis, the glycolysis reaction there. So then we start with the fructose 1,6-biphosphate. And then you'll see that on the slide, the next thing that we have is the fructose 6-phosphate. Going back to that 6-carbon sugar, the glucose 6-phosphate. And there we have the phosphate, there we have, the glucose. It's going to have the glucose that we need for energy. And so the requirement to make that glucose molecule is going to be 2 pyruvate, 4 ATP, 2 GTP, 2 NADH, and 6 waters, or 6 H2O. So aldolase is an enzyme that's used in both glycolysis and gluconeogenesis. So in glycolysis, it's going to turn that 1,6-biphosphate into two different 3-carbon sugars, which are DHAP and GAP. And then gluconeogenesis, it's going to take those two 3-carbon molecules and put them back together to make that 6-carbon molecule. Now we can use our blood serum to measure how much aldolase we have in our bodies. So in the ALT and AST test, it's going to determine the function of your liver, or if there's going to be to determine if there's a problem with your liver. If these enzymes are elevated, then that means that there may be a problem with your liver working to detoxify what comes through it. Now, the muscle tests, the CPK and the LDH, those tests are not used as much anymore, but if you measure, you can still determine if there's a problem with muscular structures in your body. Now remember that aldolase is an important enzyme, and it catalyzes fructose 1,6-diphosphate into the 2,3-carbon sugars and glycolysis. And in gluconeogenesis, it's going to do the opposite. It's going to take the 2,3-carbon sugars and going to make them a 6-carbon sugar. Now, there are 3 isozymes of aldolase. There's A, B, and C, which have been discovered so far, and it's going to be found in the cytoplasm and in the nucleus of cells. Now, there are also 2 amino acids that are involved with aldolase. They're lysine and tyrosine. Lysine is going to covalently bind, it's going to stabilize the molecule, and then tyrosine is going to be that hydrogen acceptor. Now, there are 3 types of this enzyme, aldolase, and there's A, B, and C. So A is produced in adult muscle, it's abundant there. And B is very specific to the liver, the kidney, and the small intestines. And C is found in fetal cells and sometimes cancerous cells, but it's also found in the hippocampus and Purkinje cells in the brain. Now, B is the only one, A and B are synthesized in newborns. And the adult aldolase B is the only one that is gene expressed. So we're going to be talking about the expressing of that gene and the transcription factors. So let's talk about transcription factors. First of all, let's define them. So the proteins that activate genes are transcription factors. We're going to be talking about 2 transcription factors and the enhancer binding proteins. The HNF1 alpha and HNF3, both of them have to do with metabolism. The first one is for growth and development of the beta cells that are in the pancreas that's going to make and secrete insulin. And the second one is for the metabolism of the pancreas and the liver. And also I'm going to be talking about the cat box. So just to review, the cat box is where the binding site starts for RNA transcription. If you'll look on the next side and read that slide, I thought this was expressed. I couldn't change any way that it said. I thought that was expressed very well. And it's expressed by Nerlov in 2007, his information, and you can read the complete article at the end. So what's the big deal with aldolase B? So the B isozyme is crucial for fructose metabolism and gluconeogenesis, So mutations with this genes will result in the diminished activity, which causes a fructose intolerance. It also causes hyperglycemia. It can cause growth retardation, kidney dysfunction, liver failure. If it is not treated, it can lead to a coma or a death. Now, remember that the site that it's going to bind to, it's going to signal to bind to, is the cat box. And through discussion, and we also know that the 2 overlapping binding sites are competitive. Those 2 transcription factors, HNF1 and HNF3, are competitive places where they bind. And through much discussion and much experimentation, it was found that the sites at those sites, they must bind competitively because one will stop the other one from malfunctioning and growing out of control. And we know that we see when they mutate, we see disease, and in some cases we also see cancer. So it's a complicated reaction of these two transcription factors and the enhanced binding sites and the enhanced proteins for binding. Yet it is essential for the liver to perform correctly. Thank you for listening to the presentation. I hope that you learned a little bit from this. I've had a great time with you all this cycle, and I wish you the best of luck in all of your endeavors and your continual education. Thank you.